Excuse me. All right. Hello, everyone. Welcome to our second installment of our High Elevation Five Needle Pines 2025 through 2026 Science and Management webinar series. My name is Lou Deloise. I'm a PhD student in Danielle Ulrich's lab at Montana State University. And I co-organized this webinar with two other great students, Karina Lee, a master's student in Diana Tombach's Forest Ecology Lab at the University of Colorado, Denver, and Enzo Martinelli, a PhD student in Kara Nelson's lab at the University of Montana. We have another great presentation for today, but I'd like to start by introducing our board member, Kara Nelson, who is going to share a few announcements with us. Welcome, Kara.
Thanks so much, Lou, and thanks to all of you for joining the monthly webinar series of the White Bark Pine Ecosystem Foundation. I hope all of you are aware, but in case you're not, the foundation is the go-to organization for advancing the science, management, and restoration of high elevation pines. If you're not yet a member, I hope you will consider joining. As Lou said, my name's Kara Nelson. I'm at University of Montana, a professor of restoration ecology. My students study the ecology of white bark pine, but also specifically monitoring restoration activities and impacts. I joined the foundation and then later its board because of its outsized impact on bringing our community together, thus this webinar series that it hosts. As a member, you get benefits, access to Nutcracker notes, information about conferences. But I wanted to make the point today that as a member, it's not just about the member benefits, but supporting all of the work of the foundation, which translates into on-the-ground impact for white bark pine and other high elevation pines. So at the start of the webinars, as Lou mentioned, there's some announcements from a board member. And I thought today I would focus on a new committee of the foundation, a new organization, which is the Science and Management Committee, which I have the honor of serving as its lead over the next year. And this committee includes so many activities. So the conference committee is embedded within the Science and Management Committee. We just had our conference in Bozeman with over 100 participants. Thanks to those of you who joined. There's also a genomics working group, a high five working group, the National Restoration Plan working group. There's a protocol cache, publications committee, and then of course this webinar series. So there'll be a link in the chat to join the foundation. I hope those of you who are not members will just take a minute right now, go to the website and join us and support the work of the foundation. So thanks for ceding a couple minutes to me, Lou. I'll turn it back over to you.
Great. Thank you so much for joining us, Kara. As before we start our webinar, I just want to mention that if you missed any of our previous installments, you can find recordings of the talks on our YouTube channel. The link to our website will be posted in the chat shortly. Finally, after the presentation, we will have a question and answer session where you can share all of your comments, doubts, or questions. Post them in the chat box, and our team will be collecting them and asking them to Jessica Harris. Now it is my pleasure to introduce today's speaker, Jessica Harris. Jessica grew up in a small forest community on Lake Michigan where she fell in love with five-needle pines, fungi, and rare plants, and developed a passion for protecting forest ecosystems. She obtained her bachelor's in environmental science with a minor's in chemistry and biology from DePaul University in Chicago, and in 2018 she obtained an internship and then job working in the Botanical Collections at the Field Museum of Natural History in Chicago. She then went on to work at Oak Ridge in Tennessee as an ecophysiology lab technician. Most recently, she worked with Dr. Danielle Ulrich on her master's research looking at early seedling traits in Pinus albicaulis and Pinus flexilis across climate and rust resistance levels. And with that, I'll turn it over to Jessica.
All right. Thanks, Lou, for that introduction, and thanks everyone for attending today. I'm really excited to share some of my work with you guys. I'll be presenting my Montana State University master's research titled Comparing Early Seedling Traits in Whitebark Pine, or Pinus albicaulis, and Limber Pine, Pinus flexilis, across climates and rust resistance levels, which I have completed thanks to the help of my committee members, Danielle Ulrich, Brian Smithers, and Justin Runyon. So today we're just going to go through the introduction, methods, results, discussion, conclusion of our 4 research questions. As you may know, whitebark pine or pile and limber pine or piffle are 2 of 6 high elevation 5-needle white pines that serve as keystone species in subalpine ecosystems of Western North America. This means that they have a disproportionately large impact on their surroundings despite their small abundance. And they also help maintain the structure, diversity, and functioning of an ecosystem. Pile is shown in blue here and has a more northern distribution and is found in colder, higher elevation habitats, while piffle, shown in orange, has a more southern distribution and can be found in more arid conditions, but has a broader elevational range than pile. These species have high ecological value. High five species are often the only tree species capable of surviving in the harsh conditions of the alpine ecosystem, where they establish on poorly developed rocky soils and withstand intense wind, solar radiation, and drought conditions. Due to this, pile and piffle are slow growing, but stress tolerant and long lived. They also help retain snowpack, which promotes a more consistent supply of soil moisture throughout the summer season. And they also reduce soil erosion, as well as create microclimates that support the growth of and establishment of other species. Additionally, their large nutritious seeds are really big key food sources for the primary avian disperser, the Clark's nutcracker, as well as the threatened grizzly bear, among other organisms. Despite this, both species are declining at alarming rates due to mountain pine beetle infestation, lower story species encroachment due to fire exclusion, climate change-induced drought and heat stress, and then white pine blister rust, or WPBR. Our project just focused on climate change and white pine blister rust. Climate change-induced mortality has been pronounced in high elevation forest, where warming has been occurring faster than the global average. Warmer temperatures reduce soil moisture through increased evapotranspiration rates. And this ultimately leads to widespread drought, heat stress, and overall forest mortality. This has been prompting pile and piffle to move northwards and upwards, but this has been problematic because pile and piffle have long regeneration times where their cones can take up to 50 years to be reproductively mature, and they have notoriously low establishment rates, in addition to limited gene flow. And so because of this, it's challenging for them to naturally adapt and migrate in time to combat climate change. White pine blister rust has also been considered to be the most prominent agent of mortality for pile and piffle, where they act as the primary host to the non-native fungal pathogen Crenardium rubicola. C. rubicola entered the western U.S. through Vancouver in 1906 and has since spread southwards and eastwards through the Intermountain West and through the Rocky Mountain region of the U.S. and Canada. C. rubicola forms resinous cankers that interrupts the flow of nutrients and water within host stems and branches, which ultimately leads in girdling and killing the tree. And this is especially problematic for seedling regeneration, just because it can cause adult tree crown dieback and limit cone reproduction. So, in the northern US, Rockies, and adjacent parts of Canada, white pine blister rust-induced mortality rates have exceeded 90%. And due to all of these factors, as of 2019 in the US, 54.1% of adult pile trees were found dead, and 42.3% of adult piffle trees were found dead. In Canada, pile and piffle are listed as endangered under the Species at Risk Act as of 2012 and 2014, respectively. And in the US, pile is listed as threatened under the Endangered Species Act as of 2022. Luckily, all high-five pines exhibit some degree of natural genetic resistance, albeit at low frequencies. The primary restoration strategy for pile is outplanting rust-resistant seedlings, and while piffle mortality lags behind pile, in the future, piffle may also need to be outplanted. Pile resistance exists through multiple genes, which generally suppress but do not prevent disease upon infection. And so there's a spectrum of resistance from A to F, where A is the most resistant level. And then in piffle, resistance is primarily through one gene, CR4, which if present, basically does prevent disease upon infection. And so there's several large-scale programs that essentially are aiming to identify these rust-resistant trees and plant their progeny. This process involves identifying the cones in the field, planting these seeds in the greenhouse or the nursery, exposing those seedlings to white pine blister rust spores, and then monitoring their symptoms. And then after 2 to 3 years, finally outplanting them. And so altogether, this process takes about 3 years and can cost up to $2,000 to $2,400 per hectare. So given these costs, we want to ensure that outplanted seedlings establish and survive into adulthood. Seedlings or 0 to 2-year-old individuals are inherently more vulnerable than adults due to their shallow, underdeveloped root systems, their immature above ground tissue that has minimal photosynthetic capacity, and their small stature. All of these factors make seedlings especially susceptible to drought, frost heave, wind and light exposure, and trampling and herbivory due to big guys like this fella. And so in particular, the establishment stage, which is, oops, my bad. So the establishment stage, which is when these primary leaves develop, is an especially challenging bottleneck for tree populations, especially for high-five species, where in the field mortality rates can be up to 90%. And once past this stage, though, survival generally improves. And so despite these differences in life stage and that seedlings drive species distributions, a lot of research still focuses on adults rather than the first year of life. And because of that, the physiological mechanisms of seedling establishment remain poorly understood. Okay, and so in addition to establishment, it's important that outplanted seedlings are planted in locations that will be climatically suitable now and in the future. Plant populations have evolved over time to confer higher survival and reproductive success in their climate of origin, and this is called local climate adaptation. Seen here, we have our pile seed zones, which were created specifically to identify those rust-resistant trees and find suitable outplanting locations. They were created by integrating the variation of the genetic variation and local adaptation of pile families so that we could create a larger scale of adaptation that was bigger than an ecotype or a provenance. Historically, outplanting has involved keeping seed transfer within the same seed zone. However, with local adaptations, however, with climate change, local adaptation may no longer align with current or future climate conditions. In addition to considering how important this could be, I only know of one study that has explicitly come up with outplanting guidelines outside of the home seed zone. Another aspect for successful outplanting is just ensuring that we are outplanting not only rust-resistant seedlings, but seedlings that are competitive in other ways. Undoubtedly, outplanting resistant seedlings is essential for the future of pile and piffle. But we also want to ensure that these seedlings can withstand other stressors. So by outplanting only rust-resistant seedlings, we may be unintentionally selecting for traits that are associated with resistance that could influence future forest health and forest genetic diversity. For instance, when we artificially selected for resistance in western white pine, we did lose some rare genetic alleles. In addition to that, typically in other plants, it's been found that resistance negatively correlates with growth and reproduction, which is why it's thought maybe that these resistant frequencies are found at such low percentages. But in pile and piffle, we've found that actually resistance really does have is tied to geographic variation, which may suggest that resistance is associated with abiotic stress tolerance. For instance, for pile resistance is higher in Washington and Oregon, and piffle, at least as far as we know for now, is highest in southern Colorado. And in general, high 5 species have increasing resistance with increasing elevation. And so some of these things might lead us to believe that the R gene is still around just because it's co-evolved with cold or drought tolerance. And a lot of studies have found that resistance generally does use the same proteins that are used in cold and drought tolerance. And that might be why, despite that, C. rubicola is not a non-native pathogen, resistance is here. So only a couple studies to my knowledge have directly compared resistant and susceptible groups in pile and piffle. And in these studies, generally, piffle resistant was found to have more conservative traits than piffle susceptible. And then, as stated earlier, successful outplanting might also include piffle in the future. Pile and piffle have really similar ecological roles. They're often indistinguishable without cones. They have similar seed size. Both are dispersed by the Clark's nutcracker, have similar or the same threats. However, pile is known as a specialist that occurs generally within that 1600 to 3600 elevation range. While piffle is known as a generalist that actually has a wider elevation range, as low as 870 and then even higher than pile at 3,800-ish. And so comparing pile and piffle seedling traits could help us understand why these two similar species have contrasting distributions, and also give us some insight on what the classification of generalist versus specialist means, especially in the context of climate change, just because it's thought that generalists may fare better with the changing climate. Only a few studies have explicitly studied pile and piffle seedling traits, with even few investigating differences in gene resistant type. Okay, and so what are the mechanisms of seedling establishment? So it is the ability of a seedling to obtain and utilize CO2, light, and water, as well as nutrients for photosynthesis, to grow and tolerate stress. And one way that we can measure these mechanisms is through measuring traits like phenology, growth and survival, and non-structural carbohydrates, or NSC traits. Phenology traits can provide insight on the length of the growing season, cold and drought tolerance, when to outplant. Growth and survival can give us some insight on species competitive ability and future forest composition. And then NSCs, which include glucose, fructose, sucrose, and starch, which are the products of photosynthesis, can provide some understanding of how those are being used, whether that's for plant growth, survival, or stress tolerance. And together, these traits drive seedling establishment, survival, and species distributions. And so these traits exist on a resource spectrum from conservative on the left and then acquisitive on the right. A conservative life strategy prioritizes stress tolerance over growth and is typically found in colder, drier locations, while acquisitive life strategy prioritizes growth over stress tolerance and is found in warmer, wetter locations. And so, for example, some traits that you might find with a conservative life strategy include slower growth, later phenological timing, smaller, tougher, long-lived leaves, greater below-ground biomass allocation, as well as higher starch concentrations. And so we aim to examine the physiological mechanisms of seedling establishment to improve predictions of future forest composition and species distributions under climate change. And by focusing on that first year of life in seedlings with a range of resistance levels and 2 keystone species, we could provide insight into the current outplanting practices. So we asked the following questions. Question 1, do pile traits relate to seed source climates of origin? 2, do pile traits differ between U.S. seed zones? 3, do pile traits differ between rust resistance levels? 4 had 2 sub-questions. So 4A, do pile and piffle seedling traits differ? And then 4B, do pile and piffle RNS groups differ in the same way? And so for our hypotheses, we hypothesized that traits would reflect the seed source climate of origin, whether that's for all of pile or just US pile and the US seed zones. Specifically, with those colder seedlings originating from colder, drier locations having more conservative traits, while seedlings originating from warmer, wetter conditions having more acquisitive traits. For question 3, we hypothesized that more resistant seedlings would have more conservative traits. For question 4A, we hypothesized that pile would have traits indicative of a specialist strategy. Specialists are generally either conservative or acquisitive, and considering pile's habitat, we hypothesized it would be more conservative. And piffle would have traits indicative of a generalist strategy. Generalists can have both conservative and acquisitive, and so we hypothesized that piffle would have those. And then for question 4B, we hypothesized that pile and piffle RNS groups would differ in the same way, just because pile and piffle are pretty similar species. Moving on to our methods, we first obtained pile seed from Washington and Oregon in the US, as well as from BC, Canada, and we also obtained piffle seed from Alberta, Canada. Here in this map, pile's distribution is in blue, piffle's distribution is in orange. We have our 9 US seed zones outlined here in black. Our seed zones are labeled 1 through 8, but keep in mind that zone 2 is split into 2W or 2 west, and 2E or 2 east, making it actually 9 seed zones. Our pile families are the black circles, our piffle families are the black squares. And then we have our rust resistance, where again, resistance is highest for level A, lowest for level F, and we have A in white and F in dark red. And then because piffle is resistant or susceptible, it's white or red. The size of the pie chart is the number of families that were present in the seed zone for pile or in the location for piffle. And so we had 51 pile families across 17 locations and 3 families per location. We had our 9 seed zones and we had rust resistance data, while we did not have seed zones or rust resistance data for British Columbia. These pile seed were donated by the U.S. Forest Service and the British Columbia Forest Service. We had 12 piffle families distributed among 4 locations for 3 families per location, 6 resistant, 6 susceptible, donated by the Alberta International Tree and Seed Center. And so to test our hypotheses, we grew our seedlings in a common growing environment, such as a greenhouse. Essentially, you grow all of your different groups in the same growing environment. And then if the traits differ, you can assume that it's due to genetic adaptation to the climate of origin. And if they do not differ, you can assume that the plants are basically adapting to that local growing environment, which is indicative of phenotypic plasticity. And so identifying whether it's genetic adaptation or phenotypic plasticity can help identify where to plant seedlings. So for greenhouse processing, this was done at the MSU Plant Growth Center from October of 2021 to May of 2022. This was an extensive process of processing, stratifying, germinating, sowing, transplanting, around 7,500 seeds for this project alone. Essentially, we soaked our seed in 1% hydrogen peroxide for 24 hours, aerated it in cool tap water for 4 to 8 hours. We stratified pile in a warm stratification at 20 degrees Celsius for 30 days. And then we also cold stratified both pile and piffle at 2 degrees Celsius for 90 days. Once stratification was over, we germinated them for 4 days at 25 degrees Celsius for 18-hour photo periods, and then sewed them into these containers. Okay, and so we measured 84 total traits. We had 10, 56, and 18 phenology, growth, and NSC traits. For our phenology traits, we had the proportion survived within a family to each life stage. And so these life stages were emergence, bud formation, and needle burst. Emergence was when we could see any plant tissue above the soil. Bud formation was the development of the small bud in the center of the seedling. And then needle burst is when the needle is elongated out of the bud. And in addition to those traits, we also did survival overall and then the time to each life stage. And we did this on a weekly basis for a year. For our 56 growth traits, that was divided into 28 above ground, 20 below ground, 8 whole plant. And we did this 4 times every 3 months for a year, where mostly we obtained this data from destructive harvesting and image analysis through ImageJ and rhizovision. We had really extensive statistical analysis, but all you really need to know is we did logistic regression for the phenology proportions and then linear mix effects on everything else. Okay, and now to the results and discussion. So due to having 84 traits for 4-ish questions, we decided just to focus on 48 traits per question for our results and discussion. And so to understand the climate requirements and adaptive capacity of our pile families, our first research question analyzed trait climate relationships for US and BC pile. So as a reminder, seedlings originating from colder, drier locations would be more conservative. Seedlings originating from warmer, wetter locations would have more acquisitive traits. We obtained our climate data from ORNL DAMAT from 1992 to 2021. This value was a daily value averaged over the year, averaged over 30 years. Due to high multiclinearity, we only included latitude, longitude, elevation, precipitation, snow water equivalent or SWE, maximum temperature or Tmax, and vapor pressure or VP. We used derived from human package and manual forward selection using a AIC criteria of 4 or greater to select our climate variables. And ultimately, we found that only 34 of our 84 traits, or approximately 40% of our null models, improved with the addition of a climate variable. Okay, and so first to orient you to this figure, we have our latitude going from left to right at 32 degrees all the way up to 54. And then we have our response trait on the y-axis for panels A through F. And then our locations are listed from highest latitude to lowest latitude, with these purple locations having the coolest, lowest solar radiation, and these red locations having the warmest, greatest solar radiation. So we found that this was partially consistent with our hypothesis. We had expected that temperature, or SWE, would be the most important, but latitude was actually selected for 88% of our traits. And latitude had a significant relationship with over half of those traits. In our data set, latitude could be a proxy for other climate variables. Specifically, higher latitude seedlings were found at lower elevations with a lower Tmax and lower solar radiation. And so, therefore, latitude could be representing some of the traits that we originally hypothesized. This figure generally is showing that higher latitude seedlings had greater survival at early life stages and survival overall. So in panel A, for the proportion emerged at day 30, we see a positive relationship with latitude. And for panel B, for the proportion survived at day 95, we see a positive relationship with increasing latitude. In addition to that, we also see faster growth to each stage. And so in panel C, or the time to event for a needle burst, which is the number of days to reach that life stage, we actually saw that higher latitude seedlings reached that life stage a lot faster. They had less days to get to that stage. In panel D, higher latitude seedlings had shorter seedling height. You can see that in panel D for seedling height on day 221. Moving on to panel E, we see that higher latitude seedlings had a greater root to shoot ratio on day 150. So we see a positive relationship with latitude, which is indicating that higher latitude seedlings have greater below ground biomass allocation. And then for panel F, or leaf sucrose on day 375, we see a negative relationship with latitude, where higher latitude seedlings had lower leaf and root sucrose. And so overall, higher latitude seedlings had greater survival to each life stage. They reached those stages faster. They had shorter seedling height. They had greater below ground biomass allocation, and they had lower leaf and root sucrose. I want to make it clear that any interpretations for our results are just simply hypotheses based on what I've read in the literature, and they are not like certified true facts. But if I had to interpret my results for question 1, the takeaway is that higher latitude seedlings may have local adaptations to colder, shorter days, and growing seasons. In panels A and B, we saw that there was higher survival. This could just be due to these seedlings prioritizing survival due to the harshness of their local environment. It could also be due to these seedlings having greater survival because they're adapted to having snow around for longer, which would provide more consistent soil moisture and provide insulation against extreme temperatures. Moving on to panel C, higher latitude seedlings reach those stages faster, and that could just be due to these seedlings having greater cold tolerance to maximize the short growing season. Higher latitude seedlings had shorter height and smaller stems, which could be due to limiting transpiration and cold exposure and promoting self-shading. And they also had greater below ground biomass allocation, meaning they had more of that biomass to the roots, which could imply more mechanical stress resistance against snow and wind, and penetrating through hard, cold soils. And then we also saw that there was lower sucrose both in leaves and roots. And this could just generally be due to these higher latitude seedlings having smaller plants, lower growth rates due to those shorter growing seasons, or that sucrose has been transformed into glucose and fructose to be used in cold and drought resistance. That could be cell wall stability, turgor, cell turgor, limiting the freezing resistance of water in the cell, stress signaling, and a lot more. And so all of these things considered, latitude may be a practical first step for selecting seed sources and outplanting locations. I say this because latitude is easy to obtain. It's easy to use in conjunction with seed zones, and it can also be a proxy for not just climate, but geographical trends as well. Moving on to question 2, to help identify possible seed sources to obtain seed from in the US. Our second research question, we analyze phenology and growth traits between US pile seed zones, which are shown here. Again, we got our 9 seed zones outlined in black. Seed zone 2 is still weird with the 2W and the 2E. And as a reminder, our hypothesis was really similar to question 1, but this time it was in the context of seed zones. So only 33% of our traits differed between seed zones, with seed zones 3, 4, and 5 really standing out for having high survival, specifically with the locations of Warm Spring, Mount Hood, and Colville. To orient you to this figure, we have seed zone on our x-axis from 1 to 8. You might notice C and D don't go all the way up to 8, and that's just because there wasn't information available for them at later times. We also have our response trait on the y-axis for panels A through D. And just because zones 3 and 5 had really high survival, I have outlined them in black boxes here. So one thing that was interesting is that zone 5 had really high proportion of seedlings reach emergence and bud formation and survive overall, which we can see here in panel A for the proportion of bud formation at day 30. But then when it came to needle burst, it was a little bit different, where it was the second or third highest proportion, as we can see in panel B for the proportion with needle burst at day 161. See that here. Seed zone 3 was kind of the opposite. It had really high needle burst. Actually, it had a 2 to 3 times higher proportion than any other zone, but it was second or third highest when it came to emergence or bud formation. And then zone 4 was a little more variable, having the second to fourth highest proportions. And so, okay, they have really high survival, but they also appeared to have different contrasting resource use strategies. Zones 3 and 5 had a more acquisitive strategy, while zone 4 had a more conservative strategy. Looking at panel C, specific leaf area on day 375, we see that zones 3 and 5 are generally greater than zone 4. And ultimately, they had large, thin leaves, which may promote light or CO2 capture for photosynthesis. And then when we look at panel D, we see that specific root length on day 375 had a really similar trend where zones 3 and 5 were still higher than zone 4. This high specific root length, along with other traits, show that zones 3 and 5 had these long, thin, and voluminous roots, which show that these traits might be promoting water and resource uptake in a more exploratory capacity. And then for zone 4, we see, yeah, it's a lot lower. It had thicker, denser leaves. It had thicker, denser, more localized roots. And these traits may promote cold, drought, and high light tolerance, just because having that extra layering in the leaves and in the roots can protect from cold, specifically for leaves, protect from light. And then increase mechanical stability into those hard soils and protecting from that cold. And so getting back to our hypothesis, it was somewhat supported, but not necessarily in a very clear way. Zones 3 and 5 appeared to be warmer and drier than zone 4, and we can look at panel E for our T max and panel G for our total precipitation to see that. And that might be why there was some differences in the resource strategies. And at the same time, we see that all 3 zones had pretty high vapor pressure. And they also had pretty intermediate, moderate temperatures and solar radiation. So while there might be a climatic sweet spot, it's less clear that climate is really playing the primary role here. And so could other factors be influencing what we see? I can't really say much about rust resistance in regards to this specific question, but I did find it interesting that in the literature, zones 5, 4, 7, and 3 had really high rust resistance. And then when I look at my sites, the same zones also had really high rust resistance, and they also happen to have high survival. So it appears that maybe something like rust resistance or gene flow could be influencing what we see. And so moving on to question 3, to understand how resistance varies with other traits, we examined phenology growth and NSC traits across US pile rust resistance levels, which are the same families that we looked at in question 2. As a reminder, we hypothesized that more resistant seedlings would have more conservative traits. This is only partially supported, with rust-resistant seedlings having both acquisitive and conservative traits. And so to orient you to this figure, in panels A through C, we have rust resistance level on our x-axis, which goes from A all the way to F, from left to right. A is our most resistant level in white, and F is our least resistant level in dark red. And then for panel D, we had rust resistance group on our x-axis, where the resistant group was formed through levels A through B. The intermediate group was formed through combining levels C through D, and the susceptible level was formed by combining levels E and F. And then we had a response trait on the y-axis. So compared to less resistant seedlings, more resistant piled seedlings had both conservative and acquisitive traits. These conservative traits were higher survival to each life stage, as we can see in panel A for the proportion with needle burst at day 119. And they also had tougher, more durable leaves, which we can see in panel B with leaf dry matter content at day 150. Also had the acquisitive traits of faster phenological timing, higher above ground biomass allocation, larger leaves, as we can see in panel C through leaf area on day 375, and then a higher proportion of NSC stored as sugars, and consequently a lower proportion stored as starch, as we can see through panel D with our leaf sugar NSC ratio on day 375. So why might we see these trends and what does it mean? More resistant seedlings may have traits that support competitive function depending on what you consider to be competitive and depending on the conservationist that I'm talking to. Given that these early stages of bud formation and needle bursts are extremely temperature and light dependent, more resistant seedlings may have been able to have greater survival due to having greater cold and drought tolerance. In addition to phenology, we also saw that more rust-resistant seedlings had these more tough, durable leaves, but they also had a greater leaf area, which is a really interesting combination of traits. Having greater leaf dry matter content means that you have more lignin and cellulose in your leaves, which basically increases that mechanical stress to wind and snow and extreme temperatures, as we've been talking about. But having that greater leaf area also promotes resource acquisition, making this like a really interesting combination for leaves. We also see in panel D that essentially more resistant groups had a greater proportion of sugars as NSC, which could be indicative of just having that more immediate response to environmental stress, as I've described in previous slides. And all of these traits together ultimately may provide some insight on how rust-resistant seedlings may be competitive outside of just being resistant. And then for our last question, to provide insight on why these two similar species have contrasting distributions, we compared phenology growth and NSC traits for US pile and Alberta piffle. The pile is outlined in blue and piffle is outlined in orange. And as a reminder for question 4A, do pile and piffle seedling traits differ? We hypothesized that pile would behave as a specialist and have more conservative traits, and that piffle would behave as a generalist, having both conservative and acquisitive traits. And then for question 4B, do pile and piffle RNS S groups differ in the same way? We hypothesized that they would, just because they were similar. And so orienting you to this figure, pile is on the left, and we have the resistant, intermediate, and susceptible groups. And then piffle is on the right, where we have just resistant and susceptible groups. We have our response trait on the y-axis, and then we're using that same coloring scheme that we used for question 3. So our hypothesis was generally supported, with piffle showing traits indicative of a generalist strategy and pile showing traits more indicative of a specialist strategy. Compared to pile, piffle had both conservative and acquisitive traits. Some of the conservative traits were slower needle burst timing, which we can see in panel A. For the time to event to needle burst, we can see that piffle was later than pile. This was like a really interesting finding just because we expected the opposite. But we think this might be because our piffle families were found at a lower elevation in warmer, more dry conditions. And so maybe, utilizing that whole growing season isn't as much of a priority for piffle just because of that environmental difference. And then for acquisitive traits, piffle had greater above ground biomass allocation, which coincided with lower below ground biomass allocation, which we can see through panels B through D in the leaf fruit mass ratio at day 245, the leaf mass ratio at day 375, and the root to stem plus leaves ratio at day 375. Well, it's hard to say just based on above ground biomass allocation. Having greater above ground biomass allocation could allow piffle to not only prioritize growth, but be more competitive, therefore helping it be a generalist. And then, while it's not shown here in this figure, piffle also had greater leaf sucrose and total leaf sugar and total leaf NSC estimates. And one thing that was really interesting is that piffle had almost 2 times the amount of sucrose compared to pile. So there might be something about sucrose and pitfall's response to environmental stress. It's possible that glucose and fructose are being used to synthesize sucrose, or starches being degraded to create sucrose. But outside of that, it's also possible that pitfall may be more competitive just because it has this increased long distance transport due to sucrose that could be used for a relatively more immediate stress response. And then for our last question, were pile and pitfall RNS groups differing in the same way? No, they did not. Pile RNS differed in 16 traits, while pitfall only differed in one trait. That one difference was root mass ratio in day 245. Where we see that pitfall resistant had a lower below ground biomass allocation than pitfall susceptible, which is pretty similar to what we saw in question 3. Outside of that, we saw that pile R and pile S differed, in these 3 trait examples, while pitfall did not. And these differences may be due just to differences in gene resistance type, where pile has multiple genes involved in resistance and pitfall only has the one. In addition, an interesting takeaway is that pile R and pitfall R both had high NSC sugar proportions, where resistance maybe has some relationship with high NSC sugar proportions. Okay, and so speeding through this, question 1, pile seedlings originating from higher latitudes may have adaptations to colder and shorter days and growing seasons than those originating from lower latitudes. Seed zones 3, 4, and 5 had high survival to each stage, but survival may have been driven by contrasting strategies. Question 3, more resistant seedlings have both acquisitive and conservative resource use traits. For A, pitfall exhibited traits indicative of a generalist strategy, while pile exhibited traits indicative of a specialist strategy. Pile RNS differed in 16 traits, while pitfall RNS only differed in 1. And so for future studies, there's a lot that could be improved with this. It would be awesome to include some Great Basin families, pile families, just to get that whole pile range. We could measure other physiological traits, have a higher family sample size, especially for our NSC traits, monitor these same families into the juvenile stage, and do stress treatments, look at the same things but in other high 5 species, analyze climate using growing season and dormant seasons instead of annual climate, and then comparing pile and pitfall from the same latitudinal range with the same sample size just to make it a more clear comparison. And then lastly, thanks to my committee members, all the Ulrich lab members, undergraduate helpers, the White Bark Pine Ecological Foundation, and MSU Ecology for funding support, the Darina Genetic Resource Center, the British Columbia Forest Service and Provincial Tree and Seed Center, and the Alberta International Tree and Seed Center for Seed, as well as MSU's Plant Growth Center. And thanks to everybody who was a part of my MS journey. And thank you for listening. Do you guys have any questions?
Great. Thank you so much for your talk, Jessica. I'd like to open up the opportunity for our attendants to share your comments and questions. We have about 6 minutes left, so please go ahead and type your questions or comments into the chat. And don't worry if we don't have time to go over all of the questions. Our team will send them to Jessica with your contact information for further discussion. So, Jessica, it looks like we have a question from John Van Gundy about the purpose of soaking seed in hydrogen peroxide.
Yeah, I think, well, to be honest, I got all that just from looking at all of the nursery work that's been done for pile. I think the main reason for that is I know that it helps with seedling breaking seed dormancy, but I also think it's because of preventing like fungal infections as well. But yeah, I'll be honest, I think I just kind of read that and was like, I trust it. So
Great. Thank you for clarifying that, Jessica. We also have a question from Laurel wondering if you've published any of this yet, because she would love to cite your work in a manuscript that she's working on, which I think is exciting on both of your ends.
yeah, that's really nice. I haven't published it yet, but my advisor, Dr. Danielle Ulrich, and I are working on a manuscript. So hopefully soon.
Great. Let's see. We have a couple other questions. Could you share a little bit about what the literature says on rust-resistant trait associations?
Yeah, generally reading from Vogan and Shalettle in 2015 and 2016. Those are like the two main papers I've seen that have really looked at comparing pile and or piffle rust and susceptible groups at all. So yeah, it was cool like reading their work. A lot of my work is based on reading those papers, and they did a really good job in their introduction explaining like how rust resistance essentially is related to cold and drought tolerance through pathogen genesis 10 genes. um, but it's just amazing like how much work there is involved in it, and that um, specifically in like white bark pine and limber pine, there might be like these leucine rich repeats, which are really good for basically fast, making things happen fast. And that they're associated with that. And that's why there's like such a strong connection with resistance and abiotic stress tolerance.
Great. Thank you so much for clarifying that, Jess. We have another question from Kelly Kerr, who is wondering if you've explored other climate periods, for example, 1961 and 1990, and if you think that there's a trait relationship with climate that might change with other time periods.
Yeah, that's a really interesting question. Um, because I spent a lot of time going back and forth on what time frame to use. I didn't pick the 1960 to 1990 time frame because I didn't know if it was like representing current climate change. But it could also be useful for just showing how adult trees might be behaving more than seedlings. Just because that time period really influenced like adult species distributions. So I don't know. I guess like how do I think it would be different? Um, oof. I don't know. Oh man, I don't know. But it's definitely something to keep thinking about for sure.
Great. Thank you for clarifying, Jess. We're going to take one more quick question from the chat. Sybil is wondering if you looked at seed x-rays to compare filled or empty in embryo size, and if that was factored in terms of seed quality, which might have determined emergence at day 30.
Yeah. So this is definitely something I wish I could have done. Um, so we weren't able to actually really do anything scientific to test the seed class. I know that our US pile had like a range of seed classes. Um, I don't believe that we had that information for the British Columbia or Alberta Piffle seed groups. So it kind of made it hard for us to like, I guess, see how seed class was really affecting everything. We could see how it was affecting like the US, and I didn't see any relationships with seed class. Um, but I definitely feel like that's something that like totally could influence what we saw. And I do think it influenced our piffle seed for sure, because it didn't do super great, which we weren't expecting that. So yeah.
Great, thank you. And unfortunately, we are out of time, but I want to, on behalf of the White Bark Pine Ecosystem Foundation and the webinar organization team, thank Jessica for her time and willingness to participate in this webinar. Our next webinar series will be November 18th, featuring Josh Beisel, a PhD student at the University of Montana, presenting on how site conditions constrain the influence of fire on regeneration of whitebark pine. And thank you to everybody for attending, and we hope that you stay tuned for our upcoming installments.